and welcome to the Amplifying Scientific Innovation podcast. I'm your host, Dr. Sophia Onye, founder and CEO of the Sophia Consulting Firm, a life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast is to showcase scientific innovation stemming from global life science companies through conversations with senior leaders who share their unique leadership journey, corporate vision, and industry outlook. My guest today is Dr. Bruce Levine, a Barbara and Edward Netter Professor in Cancer Gene Therapy and the founding director of the Clinical Cell and Vaccine Production Facility at the University of Pennsylvania. He is also the co-founder of Tmunity Therapeutics, a Penn spinout. Amongst many significant accomplishments, Bruce led the first in-human adoptive immunotherapy trials, including the first use of a lentiviral vector, the first infusions of gene-edited cells, and the first use of lentivirally modified cells to treat cancer. He received a BA in biology from Penn and a PhD in immunology and infectious diseases from Johns Hopkins. Bruce has overseen the production, testing, and release of 3,000 cellular products administered to more than 1,200 patients in clinical trials since 1996. In addition, he's also the co-inventor of the first FDA-approved gene therapy, commercially known as Kymriah, which is a chimeric antigen receptor T cell, or CAR-T, for leukemia and lymphoma that is licensed to Novartis. Bruce is a co-inventor on 27 issued U.S. patents and the co-author of more than 180 manuscripts and book chapters. He's also the recipient of the William Osler Patient-Oriented Research Award, the Wallace H. Coulter Award for Healthcare Innovation, serves as a president of the International Society for Cell and Gene Therapy, and on the board of directors of the Alliance for Regenerative Medicine. Bruce and I connected on LinkedIn following his remarkable virtual panel presentation at an Esconomy Philadelphia digital event. Moreover, I have always been a fan of his transformative work in the field of immuno-oncology. Welcome to the show, Bruce.
Well, thank you, Sophia. That's very kind of you. It's a pleasure to be with you, albeit virtually. But we're adjusting, and we do what we need to do to advance the field.
Absolutely. Thank you for being a guest on the show. It's an honor and a pleasure. So my first question is my favorite because you can answer it however you choose to. And it's, what is your definition of scientific innovation?
Yeah, well, innovation is forging new paths, going into the unknown. When I was growing up, it was the Apollo era. And we were exploring new worlds and the moon and what was outside of the Earth orbit. So it's beyond replication, right? It's exploring what we need to explore to be able to devise new therapies for patients.
That is a great definition. And I think that what goes along with parts of forging new paths and going into the unknown is bravery. You have to be quite courageous to do those things. And obviously you've done that, you've demonstrated that your whole career. And I'll be curious to know what you consider to be your single most significant professional or scientific accomplishment today.
Yeah, so maybe a little bit of stubbornness or a lot of stubbornness. When I provide advice to people who want to go to graduate school, I advise them not to do it. And then if they ignore me, they must still want to, which you need persistence. And that's why they call it research, right? It's not search, it's research, which means that you have to do it a number of times. So asking for a single discovery or accomplishment is like picking favorite children. It's not something that you can really do, but certainly we've been able to translate a number of first-in-human clinical trials, including those that you've listed. And it's challenging to do in phase 1 trials, and we did that with lentiviral vectors and gene editing and other things, but it's a whole different experience when you're looking to move beyond the phase 1 trials to the phase 2 multi-center trials, and then to work with a partner to achieve FDA approval. So certainly I would mark what has been a huge team achievement within the Center for Cellular Immunotherapies at Penn, Children's Hospital, work of Carl Ju, my mentor, and working with Novartis to achieve FDA approval for Kymriah. But we're not done. And there's a lot of things that we're working on with Tmunity and with other partners to move beyond hematologic malignancies into cell and gene therapies for solid cancers, for example.
I think that that's a remarkable statement, Dr. Levine. I actually first heard about you around the time of the Kymriah approval, so I think that's a brilliant job. Well done. But then it led me to think about the beginning. I'm always concerned or curious about how things start. So why did you choose to research cellular immunotherapies?
Yeah, so, it's funny. And there was a segment on one of these news magazine TV shows way back when I was younger, before I had gray hair. And it was about bone marrow, then it was called bone marrow, now stem cell transplantation for patients with cancer. And I found it fascinating that you could take someone's immune system out of their body, put it in a freezer, give them high dose intensive chemotherapy, and basically save their life with what you had in the freezer. So I had that in the back of my mind. And then when I was in undergrad at Penn, I asked a couple of my professors, what do you see as the up and coming field for the next 20, 30 years? And I got the answer back, immunology. Okay, so then I applied to PhD programs in immunology. I attended Johns Hopkins program in immunology and infectious diseases. And in between undergrad and grad school, I had worked at Children's Hospital of Philadelphia. And it struck me, I was going to the lab to do my work. On the elevator, there would come children in wheelchairs and stretchers and gurneys, and it really put the inspiration into me to want to do translational research. And so my work at Johns Hopkins was in a laboratory in the bone marrow transplantation research laboratories located in the hospital. And so while I was doing my thesis work, I read papers by a guy named Carl June, applied to his laboratory for a postdoctoral fellowship. He accepted me there in 1992. And I began working on systems to grow T cells in the lab. We had something that worked extraordinarily well, much better than existing systems. And at some point, a couple of years into the fellowship, he said, well, how would you like to start a laboratory that grows cells for an adoptive immunotherapy trial? And so that, to me, seemed like the culmination both of my education and the inspiration that I had achieved along the way.
Wow, that is brilliant. I think it always starts with a simple idea and then you build on it. And before you know it, you become you.
Yeah.
But I'll be curious to know now if you could provide a top-line overview of ongoing work in CRISPR and CAR-Ts and that you're doing at Penn and its implication for innovative patient care.
Sure. So we have the first generation of CAR T cells approved in hematologic malignancies. But in order to make this more widely accessible to patients with cancer and other diseases, we need to work on a number of things. And I'll just list two. There are many, but improvements in manufacturing that will reduce the cost. That includes automation. It includes other improvements so that we can manufacture more of these and again, move beyond North America, Europe, Japan, and Australia to all parts of the world. And the second thing that I'll pick out of the many is improvements in potency. We have patients that respond, extraordinary success stories, but we also have patients that don't respond and relapse. So how can we change that? How can we improve the potency of the CAR construct, working with the T cells, and then moving into solid cancers? They're much more immunosuppressive on balance than hematologic malignancies. So what extra modules can we add into the CAR construct to make those T cells resistant to those immunosuppressive effects? And then once we make them more potent, then that feeds into making them more accessible.
Wow, I really like the focus on improvement in manufacturing as well as improvement in potency and tying it back to widespread access beyond just the rich countries. I was born in a developing country in Nigeria, and I always like when I speak with people like yourself, senior leaders in the field that have that global view on the advancement of therapy. So thank you for sharing that.
Yeah, we do. In fact, we've worked with investigators in Costa Rica, and I serve on the scientific advisory board of a company developing CAR T cells in India. So it's important to go beyond the first level and think of next steps. And one other thing I'll mention quickly is so-called allogeneic or off-the-shelf therapies. So we're working on those, and there are companies developing those. There's the immune system has a several 100 million-year head start on being able to treat all patients with one product. That's why I can't give you my liver, right? So we have to work on ensuring that those off-the-shelf therapies are both potent and they can be given to many patients.
That's wonderful. So thinking philosophically, how do you think the biotechnology industry has changed in the past 15 years? And along those lines, what are some loopholes?
Yeah, so there has been huge change that we've seen, and I'll take it just the past 10 years. Well, we can go back. Do you know that the very first CAR T cell clinical trials were conducted by industry, a small company called Cell Genesis that was in San Francisco? And we worked with them in the late 90s, and we showed safety and feasibility, and there was a hint of something happening, and this was an HIV. And then we started in cancer preclinically in 2004, treated our first patients in 2010. When we published the first 3 patients with chronic lymphoid leukemia that we had treated, we published in 2011. The landscape changed because at that time there was maybe one company, Kite, or the progenitor of Kite, that was developing T cells, maybe a couple others at that time, but really under the radar and looked at as a boutique, and a lot of skepticism. 2011, we published in New England Journal of Science and Translational Medicine, and companies of all sizes approached Penn to license the technology, including Novartis and a couple other very large companies. So we signed with Novartis. When that became public in August 2012, the level of interest, piggybacking on the scientific reports from 2011, really catapulted a huge increase in investment. We had Juno formed. There were other companies that were formed, others in existence, like Kite saw a surge in interest in investment. So I think it was a rising tide lifting all boats. And now we have a couple hundred companies in the CAR T or CAR NK space. It's boggling to think of that and the continuing investment. And the ripple effect we've seen is not only in the biotech and biotherapeutic companies, but it's in the tools and equipment makers. Those companies have also devoted much more resources. They've had investment themselves, and that has facilitated the innovation in the field.
Right. And it ties back to your previous definition of innovation along the lines of forging new paths and going into the unknown. So thank you for sharing that. The follow-up question is one that is fairly obvious, right? But I would love to hear your perspectives on that. What could we have done differently as an industry to mitigate the impact of COVID-19?
Yeah, so I think, look, the industry and those of us in academia were at the mercy of the infrastructure, the public health infrastructure and the government policies. But setting that aside, it really comes down to redundancy in terms of personnel, facilities, and supply chain primarily. So when we think of the impact on COVID, it's people working from home. How will you continue your development and manufacturing and testing? Well, okay, work in shifts. When we think of the supply chain, you have masks and face shields and gloves for working in the clean room.
Mm-hmm.
Do you have backup? Do you have secondary suppliers? So those things we can work with, but then in thinking of the conduct of clinical trials, if clinical sites in New York are shut down,
Mm-hmm.
then that has a big impact on the conduct of a clinical trial. If clinical sites elsewhere in the country are shut down, then that also has an impact. And then that gets to the behavior of the subjects who are enrolling in the clinical trial and even patients who would come in for therapy. So it's very complex and multifactorial problem that we're dealing with, and some countries are dealing better than others, some sites are dealing better than others, but I think in the end it comes down to uh, uh, being prepared and having redundant systems in place.
Well, thank you for sharing that. I'll be curious to know along those lines, is there any technology or company that you're currently excited about? And it could be, of course, in the cancer immunotherapy space or in the COVID-19 space or even something outside of that.
Yeah, well, there's many, and I'm not able to provide investment to anyone, but I'm really looking at some of the allogeneic technologies and iPSC cells. So there was a publication from my IACT colleague, John Rasko, on mesenchymal stromal cells produced through iPSC technology to potentially treat graft versus host disease. If we have iPSCs to produce CAR T cells or other cells or regenerative medicine products, that makes a huge impact. Now, that technology is at the stage that CAR T cells were in the late 90s, early 2000s. So it's not a next year approval technology. I think we're looking at the 5 to 10 year horizon, but that is exciting technology. Again, you ask a question like picking favorite children. There's so much to be excited about in the field, and as president of the International Society for Cell and Gene Therapy, I get top level view of that. And what I would say to those listening is get involved, recruit others to become involved in this field. It's a very exciting time.
No, I like how you tied back allogeneic stem cell therapies with your earlier point around improvements in manufacturing and potency. I think that that could probably be a step in the right direction to improve some of the efficacy rates that we have, as well as access to these important new therapies. So my follow-on question, I guess, will be when you think broadly about the industry, and obviously you're not giving any investment advice, just your opinions, what are some key consideration factors that you think will be important for sustaining innovation in the life science industry?
Yeah, so I think we've got to get over this hurdle of COVID-19, and we're going to be in a new normal, not the old normal. So I think that's an immediate hurdle to have companies in the industry and clinical trials enrolling at full speed. I think another hurdle that we haven't yet talked about is the hurdle of the regulatory environment. And we look at regulations, whether it's FDA or EMA or TGA or some other region, those regulations and accompanying guidances were written in large part in the era of small molecules and even biologics for antibodies. This is different. It's a lot number of one for the autologous products. There are patients who are very ill. So how do we think about critical quality attributes in context of the regulations? How do we think about clinical trial design? And even the staffing at these agencies, they're hiring because they're reviewing more protocols, they're reviewing more applications. So again, how can we help them as a field? And it's educating each other, and it's bringing young people into the field of immunology, cell and gene therapy, regenerative medicine, going undergraduate and even into the high schools, reaching out and educating students and the lay public about the field, and also gene editing and the implications of gene editing.
Yeah. Do you think these therapies are too complex for the public to digest, or how can we approach it differently so that people understand the importance of gene editing, for example?
Well, they are complex, and in thinking about gene editing, when I get a question on gene editing and ethics and public understanding, I ask the question back to the audience: how many of your family members and friends can explain what gene editing is to you? So there's a lot of confusion on this technology, what we're doing, the difference between gene therapy and gene editing. Gene therapy, where we might add a gene construct to a cell, or we might administer it directly. Gene editing, where we may edit a somatic cell, and how we should not edit a germline because that's changing the species. So it is very complex, not only scientifically, but from a policy and ethics perspective. And if you ask, is it possible? I think that it's possible. I certainly hope that it's possible.
Yeah.
And what we need to do is engage not only ourselves as scientists and experts, but the lay public and patients. And patients often are our strongest voice for the technology, and putting it in the context of a story where the public can be engaged.
No, that's actually what my mission for this podcast is, to help in that scientific storytelling, because these are complex ideas. We can simplify, but we cannot dilute it. And I feel that's where some of the challenges come about. But thank you for really that elegant discussion so far. My final question for you is, do you have any other commentary or thoughts? The floor is completely yours.
Well, the closing thought I always give is a thank you to our patients for enrolling on our clinical trials. They place their faith and their hope in us, and we owe them our best efforts to make progress and to be transparent with what the science is, what the results are, and how we can move the field further and farther. And I think it's truly a partnership, and it's a partnership with the public, because the public, in some way, through taxes, funding government agencies who provide grants, or through philanthropy, or through even stock ownership in companies, ultimately it's the public that is funding science. So we do need to engage together. And that's why I'm so thankful that you're doing these interviews and engaging the public. So thanks to you, Sophia.
It's my absolute pleasure, Bruce. Thank you for your time, and I look forward to staying connected with you. And thank you again.
All right, very good. Thanks.